Hello students, welcome to Read MedPrep Academy channel. Today in Classical Genetics Part 2, we are going to talk about Mendelism. The contributions of Mendel to genetics is called Mendelism. It includes all concepts brought out by Mendel through his original research on plant hybridization. Mendelian genetic concepts are the basics to modern genetics today. Gregor Johann Mendel is called the father of genetics for his various contributions. The first geneticist, Gregor Johann Mendel, unraveled the mystery of heredity. His life period is between 1822 to 1884. He was born on 22nd July 1822 in Hinzendorf, Silesia, Austria. Now it is called Hinzais in Czechoslovakia. He was born in a peasant farming family. After school education, he studied botany, physics and mathematics at the University of Vienna. He then entered a monastery of St. Thomas at Brunn in Austria and continued his interest in plant hybridization. In 1849, Mendel got a temporary position in a school as a teacher, and he performed a series of elegant experiments with the pea plants in his garden. This is St. Thomas Abbey in Austria, where Gregor Mendel joined. In 1856, he started his historic studies on pea plants. In 1856 to 1863 was the period of Mendel's hybridization experiments on pea plants. Mendel discovered the principles of heredity by studying the inheritance of 7 pairs of contrasting traits of pea plant in his garden. Mendel crossed and catalogued 24,034 plants through many generations. His paper, entitled Experiments on Plant Hybrids, was presented and published in the Proceedings of the Brunn Society of Natural History in 1866. Mendel was the first systemic researcher in the field of genetics. How Mendel was successful? He applied mathematics and statistical methods to biology and laws of probability to his breeding experiments. He followed correct scientific methods and kept accurate and detailed records that include quantitative data of the outcome of his crosses. His experiments were carefully planned. He used large samples. The pairs of contrasting characters, which were controlled by factor called genes, were present on separate chromosomes. The parents selected by Mendel were pure breed lines, and the purity was tested by self-crossing the progeny for many generations. Mendel's experimental system chose the garden pea as a sample. Why he chose pea plant? Because it is an annual plant and has clear contrasting characters that are controlled by a single gene separately. Self-fertilization occurred under normal conditions in the garden pea plants, and Mendel used self-fertilization and cross-fertilization. The flowers are large, hence emasculation and pollination are very easy for hybridization. What are the steps he did in cross-pollination of the pea flowers? He removed the male sex organ containing the pollen from a purple flower and removed it. And he took the pollen of a white flower and transferred it to the pistil of the purple flower. So the purple flower acted as the female sex organ containing flower, and the white flower acted as a male flower, and the fertilized pistil gets mature and becomes pods. Seeds in each pod are ready to be planted, and all the offsprings that are obtained in the F1 generation are purple. Mendel's theory of inheritance, known as the particulate theory, establishes the existence of minute particles or hereditary units of factors, which are now called as genes. He performed artificial pollination or cross-pollination experiments with several true breeding lines of the pea plants. Here you can see in this image how artificial pollination was performed. He took a flower containing the male and the female sex organs. He removed the anthers by cutting off the stamens which contain the pollen grains, thereby making the flower a female flower. And he took another flower from which the pollen was taken, and this pollen was transferred to the flower from which the stamens had been removed. Thus, he did artificial pollination. Now, what is a pure breeding strain? A true breeding lines or pure breeding strains means it has undergone continuous self-pollination, having stable trait, inheritance from parent to offspring. For example, the concept of true breeding is a purple flower is self to form a purple flower containing plant. And again, when it is self, it produces a pure breeding line only containing the purple flowers. Similarly, the white flower containing plants were self to form the white containing flowers. Similarly, again the white containing flowers were formed by pure breeding lines. Homozygosity. Matings within the pure breeding lines produce offsprings having special parental traits that are constant in inheritance and expression for many generations. So the pure line breed refers to homozygosity only. Fusion of the male and the female gametes produced by the same individual, that is the pollen and the egg, are derived from the same plant, is known as self-fertilization. Self-pollination takes place in Mendel's peas. Why peas was selected? Because of its true breeding character by self-pollination during repeated generations. Here in this flower you can see the flower containing the male sex organs having the anthers and the pollen grain, and the female sex organs, the ovary containing the ovules and the style and the stigma. The pollen is transferred from the flower to the stigma of the same flower. This is a common phenomenon in the pea plant. Now the experimenter can also remove the anthers, otherwise called emasculation, before fertilization, and transfer the pollen from another variety of the pea to the stigma of flowers where the anthers are removed. Here you can see how emasculation is done. It is only the removal of the male sex organ of the plant, like the cutting of the stamens that contain the anther which has the male gametophyte, the pollen grain which contain the male gametes. So the parent is removed of the stamens. This is called emasculation. And the pollen from another flower is taken and dusted on the flower from which the stamens have been removed. This results in cross fertilization, which leads to the creation of hybrid varieties with different traits. Mendel's work on the study of the pattern of inheritance and the principles or laws formulated now constitute Mendelian genetics. The first model organism taken in genetics by Mendel was the garden pea. The botanical name is Pisum sativum. 7 characters were studied by Mendel of his plant in his experiments on genetics. The 7 characters that were studied are, you can see in the table, the character, what is the dominant trait and what is the recessive trait. The stem length is the character. The dominant trait is the tall and the recessive trait is dwarf. Pod shape, the dominant trait is inflated and the recessive trait is constricted. Seed shape, the dominant trait is round and the recessive trait is wrinkled. Seed color, that is the cotyledon color, yellow is the dominant trait and the recessive trait is green. The flower position, axial position of the flower is the dominant trait, whereas the recessive trait is the terminal position of the flower. The flower color, the purple color of the flower in the pea plant is taken as a dominant trait and the recessive trait is the white flower. The pod color, the green is a dominant trait and the recessive trait is yellow. So the 7 characters of Pisum sativum have their genes in the chromosomes. For example, the plant height, the gene is capital L and small e, and the dominant trait is tall and the recessive trait is dwarf. In seed shape, the gene is represented as capital R. The dominant trait is round and the recessive trait is wrinkled. In the cotyledon color, the gene is taken as I. The dominant trait is yellow and the recessive trait is green. In the flower color, the gene is capital A. The dominant trait is purple and the recessive trait is white. In pod color, the gene is capital G and capital P. The dominant trait is green and the recessive trait is yellow. In the form of the pod, the gene is capital V, the dominant trait is inflated and the recessive trait is constricted. In the flower position, the gene is capital F and small a, the dominant trait is axial and the recessive trait is terminal. The 7 characters in the garden piece are shown in the plant's 7 chromosomes. If you take the chromosome number 1, the upper arm shows the flower color and the lower arm shows the seed color or the cotyledon color. The second and third chromosome are not specified with any characters. The fourth chromosome, the upper arm shows the pod shape, the plant height, and the lower arm shows the flower position in the chromosome number 4. In chromosome number 5, the pod color is represented in the upper arm. The 6th chromosome does not show any character, and the chromosome number 7 shows in the upper arm the seed shape. So Mendel worked at the rules of inheritance and arrived at the correct mechanism before any knowledge of cellular mechanism, the DNA, the genes, the chromosomes became available. Mendel's insights and meticulous work into the mechanism of inheritance played an important role, which led to the development of improved crop varieties, and a revolution occurred in the crop hybridization. Mendel died in 1884. In 1900, the work of Mendel's experiments were discovered by 3 biologists: Hugo de Vries of Holland, Karl Correns of Germany, and Erich von Schermack of Austria. The molecular answer to understand the Mendel's gene function for regulating the white color in peas found a solution to the genetic mystery of Mendel's white flowers. It is quite fascinating to trace the Mendel's genes. After many years, in 2010, the gene responsible for regulating the flower color in peas were identified by an international team of researchers. It was called P gene A, which was present in chromosome number 1, which encodes a protein that functions as a transcription factor, which is responsible for the production of a pigment called anthocyanin. So the flowers that contain the pigment anthocyanin are purple. So pea plants with white flowers do not have anthocyanin, even though they have the gene that encodes the enzyme involved in the synthesis of anthocyanin pigment. Here you can see a purple flower on the left side of pea with the P gene A. On the right side you can see a white flower of pea which does not contain the P gene A. What is the mechanism of dominant and recessive flower color? The dominant allele is responsible for the production of the functional enzymes, which causes the production of anthocyanin, which gives the flower the purple color, which is a dominant character. Whereas the recessive allele produces no enzyme. So it's a non-functional enzyme. So there is no production of anthocyanin. Since there is no production of anthocyanin, the color of the flower is white, and this character is recessive. Researchers delivered normal copies of gene A into the cells of the petals of white flowers by the gene gun method. When the gene A entered in a small percentage of cells of the white flowers, it is expressed in those particular cells accumulated anthocyanin pigments, and they became purple in color. Here you can see the white flowers on the left side. When the introduction of the gene A occurred, they become purple in color because of the production of anthocyanin. In white flowers, the gene A sequence showed a single nucleotide chain that makes the transcription factor inactive. So the mutant form of gene A do not accumulate anthocyanin, and hence they are white in color. Mendel noticed 2 different expressions of a trait. Example, tall and dwarf. Traits are expressed in different ways due to the fact that a gene can exist in alternate forms or versions for the same trait, and these alternate forms of the expression of the genes is called alleles. Here you can see the dominant trait character of a tall plant, which is represented as capital T, whereas the recessive trait, which you can see in the dwarf plant on the right side, is represented as small t. If a diploid individual has 2 identical alleles of a gene at a given locus, it is called homozygous. If it is dominant, it is called homozygous dominant, capital T. If it is recessive, it is called homozygous recessive, small t. A diploid individual with two different alleles at a specific gene locus is called heterozygous. It is represented as capital T and small t. Mendel's non-true breeding plants are heterozygous and they are called hybrids. The superiority of a hybrid over either of its parents in one or more traits is the heterosis or hybrid vigor. What is a gene? The determinant of a characteristic of an organism. Otherwise, it was called Mendel's factor. Gene is symbolized as underlined or italicized. What is a genetic code? The set of 64 triplets of bases or codons corresponding to 20 amino acids in proteins and the signals for initiation and termination of the polypeptide synthesis is called the genetic code. What is a locus? The site or position of a particular gene on a chromosome is called a locus. What is a genome? The total constitution of the genes contained in a cell is called a genome, or the genetic constitution of an individual is called a genome. When the gene has two alleles, the dominant allele is symbolized with the capital letter T and the recessive allele is represented with a small letter t. Capital letter T is for tall and small letter t is for dwarf. When both alleles are recessive, the individual is called homozygous recessive, small t, and that forms the dwarf pea plants. An individual with two dominant alleles is called homozygous dominant, capital T, which forms the tall pea plants. One dominant allele and one recessive allele, capital T and small t, denotes non-breeding tall pea plants. They are called heterozygous tall. Here you can see the genotype, homozygous tall or dominant, represented by capital T. Dwarf plants, which is homozygous recessive, is represented by small t. Heterozygous tall plants are represented by capital T and small t, since the dominant allele capital T is present. The tall allele is stronger than the dwarf allele. We say it is dominant. The dwarf allele is weaker than the tall allele. We say it is recessive. Mendel proposed two rules based on the observations of monohybrid cross. Today, these rules are called laws of inheritance. The first law is the law of dominance and the second law is law of segregation. These scientific laws play an important role in the history of evolution. Law of dominance. What is this? The characters are controlled by discrete units called factors which occur in pairs. In a dissimilar pair of factors, one member of the pair is dominant and the other member of the pair is recessive. Here you can see the phenotype, the color of the fruit, and the genotype. Dominant is capital A, and recessive is small a. Whereas the heterozygous dominant is capital A and small a. This law gives an explanation to the monohybrid cross. The expression of only one of the parental characters in F1 generation and the expression of both in the F2 generation. It explains the proportion of 3:1 obtained at the F2 generation. Here you can see the parents. The tall plant capital T is crossed with the dwarf plant small t. The gametes produced by the tall plant is capital T and the dwarf plant is small t. And when they are crossed in the F1 generation, the output is heterozygous tall. All the plants are tall. They are heterozygous. When the heterozygous tall plant is crossed with the homozygous dwarf plant, the output obtained is 3:1. That is, 3 plants are tall and 1 plant is dwarf. And out of the 3 plants that are obtained as tall, 1 is homozygous, 2 are heterozygous tall, and the other plant which is obtained is a dwarf homozygous plant. Now, what is law of segregation? This is called law of purity of gametes. Alleles do not show any blending. Both characters are seen as such in the F2 generation, although one of the characters is not seen in the F1 generation. During the formation of gametes, the factors or alleles of a pair separate and segregate from each other, such that each gamete receives only one of the 2 factors. Here you can see the alleles that separate during gamete formation. In the F1 generation, you can see the heterozygous plant, capital T and small t, produces two types of gametes, capital T and small t. So the gametes segregate independent of each other. And in F2 generation, the 3:1 ratio is obtained as we explained earlier. A homozygous parent produces similar gametes, and a heterozygous parent produces two kinds of gametes, each having one allele with equal proportion. Gametes are never hybrid. Here you can see a yellow colored pod producing the gamete Y and the green color pod producing the gamete small y. And the fusion of the capital Y and the small y causes the hybrid plant heterozygous capital Y and small y, having, since Y is dominant, having yellow pods. In the F1 generation, when two heterozygous plants capital Y, capital Y small y and capital Y small y are crossed, they produce two types of gametes, capital Y and small y. You can see according to the checkerboard where the three types of yellow pod containing plants are obtained and one green pod plant is obtained. Out of the four plants, the three plants are yellow pods and one plant is green pod. And out of the three yellow pod plants, one plant is homozygous. Two plants are heterozygous. So today in Classical Genetics Part 2, we discussed about Mendelism. Thank you. Kindly subscribe, like, share and comment the channel Read Med Prep Academy. Log on to www.readmedprepacademy.com. Our Facebook ID is Read Med Prep Academy. Our email is readmedprepacademy@gmail.com. Kindly post your questions in the comment box. We will reply with appropriate answers. Have a nice day. Thank you.